Okay, hello, my name is Corey Keene. I'm currently a postdoctoral scholar at UCSF, and today I will be giving a lecture on microbial stains with a focus on the Gram stain. Gram stain is a very important stain in microbiology. The students are going to be required to obviously know how to do this technique, and it's very important in identifying student unknowns in later parts of courses, of microbiology courses that I've taught as well. I usually like to start my lectures with a little bit of comical humor to kind of get our students engaged and to let them know when our lecture is starting. And this is a good example of a gram-positive, gram-negative joke, as many of those as you can find, at least on the internet, where a gram-positive cell is controlling the gram-negative cell. So the outline today, we'll start by going into what bacterial stains are used for, and then we'll delve into a little bit about the Gram stain, the background of the Gram stain, working into what the Gram staining protocol, how that's actually done, and where we can troubleshoot things in the Gram stain that require, or if there's something that happens while the students are practicing in the lab and they don't have consistent results, that we can talk about this here so that we can give them some ideas of how to troubleshoot their own results. Okay, so there are multiple ways to stain microbes in a lab setting. Most often, if we're just looking to identify structure, we can start with the Stigl stain, just using either crystal violet or some other type of stain, methylene blue. Or we can use differential stains, which take advantage of characteristics that different bacteria have, such as the Gram stain shown here above. We have gram-negative rods and gram-positive rods in the same area, or an acid-fast stain, or we can actually use special stains that help us identify whether or not bacterial cells have other special properties, such as capsules or flagella in this case. So today we're going to be focusing on this primary differential stain, the Gram stain, because it's one of the most important ones that's used in our lab setting. It's one that you will be doing almost on a daily basis, and it's one that is very important to have down before these students get into doing their unknown projects. Excuse me. So a little background on Gram stain. It was invented by Hans Christian Gram, who originally was using this as a technique to try to visualize bacteria a little bit better in sections of lungs so that they can better identify whatever cultures a patient could be infected with. But they ended up finding out that this had a useful property, and that it was able to distinguish one type of cell versus another type of cell. And you know, today now, this particular technique is now one of the most utilized stains for microbes. It's very important, excuse me, it's used clinically and diagnostically to tell whether or not you have a particular type of gram-positive or gram-negative organism. And again, this helps to differentiate microbes by the presence of peptidoglycan in the cell wall. Gram-positive cells are going to have a very thick layer of peptidoglycan versus gram-negative cells that are going to have a very thin layer sandwiched between two lipid bilayers in the bacterial cell. And this is best represented by this cartoon. It's a little bit complex, so we'll walk through it slowly here. So on the top here, you have your gram-positive bacteria. It has this thick layer of peptidoglycan, and there's also some other sugary substances that are present in your peptidoglycan that have other types of functions that we're not going to really talk about today. And then under that, you're going to have your cell membrane for your bacterial cell. Okay, so this composes a gram-positive bacterial cell wall. This gram-positive cells, examples of gram-positive cells include Staphylococcus aureus or Bacillus megaterium, and these are cultures you'll likely see in the lab. For gram-negative cells, you can see it, they're a bit more complex, but the basic idea is that you have a lipid bilayer on the top and a thin layer of peptidoglycan under that first lipid bilayer, and then under that you have your cell membrane, and this again composes the cell wall of the bacteria. Examples of Gram-negative bacteria include Escherichia coli or Neisseria gonorrhea. And again, we'll kind of come back to some more examples of this later. But again, the take-home message of this is that gram-positive cells have a thick peptidoglycan wall. Gram-negative cells have a thin layer of peptidoglycan. So the gram staining protocol itself is very easy. The process, you take the cells from either a tube or a colony. You take them from their source, and then you streak them on your slide. From there, you're going to take and heat fix your bacterial cells to the slides to ensure that they don't wash off from subsequent stainings. And we probably have covered heat fixing by this point, but you basically take your slide, run it under a Bunsen burner for about 5 seconds, going back and forth so you don't burn your hand. Once you have your slide fixed, you'll then stain with crystal violet, and then with iodine, rinse off the crystal violet and iodine with ethanol, and then stain with safranin as a counter stain. And we'll go through this step by step here in a minute, but this is just basically the summary of what we're going to be doing. The results from this would be if you have a gram-positive cell, your slide will appear purple, versus a gram-negative cell, your cells are going to appear pink. So what's actually happening? Let's break this down a little bit. So how do we get to our purple versus our pink cell? Okay, so after you've heat fixed, you're ready to stain, you're going to start with crystal violet, and everything that has a peptidoglycan wall is essentially going to take up this crystal violet, and everything on your slide is going to appear purple if you were to look at it under a scope. Okay, so next, you'll wash off that crystal violet, and then you'll stain with iodine, which again is a mordant that helps basically bind any stain with the peptidoglycan layer that's going to be present there. And since gram-positive cells have a very thick layer of peptidoglycan, they're going to hold on to that stain much better than those of the gram-negative cells. Okay, so even though you have this going on, the iodine binding, the iodine helping the crystal violet bind with the peptidoglycan, everything is still going to appear purple. Okay, it's not until you actually add ethanol where things get a little rough, and we'll talk about what can happen in troubleshooting mechanisms for this a little bit later. So once you have your gram-positive cells and your gram-negative or your gram-positive, gram-negative samples, you're going to drip ethanol on the slide until all of the crystal violet runs off. And what will happen is because your gram-positive cells have a very thick layer of peptidoglycan, they're going to retain that purple color. The gram-negative cells are all going to have whatever staining they had with the crystal violet wash out, because that peptidoglycan layer is so thin, it's going to basically take everything with it and leach all of the color out. Okay. But now you're going to have a slide that has gram-positive cells and cells that appear to have no color. So you need to be able to visualize those cells somehow. And that's what we use essentially the safranin for, is you're going to take the safranin, counterstain both slides, and your gram-positive cells, which already are well stained with crystal violet, aren't going to take up the safranin, and they're going to retain the purple color, where your gram-negative cells are going to essentially appear purple now because they don't have any crystal violet, because it's all been leached out. Okay, so that's kind of the summary of a gram stain. So what are some things that can happen here? Okay, so the tricky step is the ethanol decolorization step, because if you wash for too long, you can have certain things that happen. So a proper rinse, you'll have your gram-negative cells appear totally clear, but your gram-positive cells will appear purple, or your gram-negative cells will appear totally clear. But if you've over-rinsed, some of your gram-positive cells will also appear clear, and your gram-negative cells will also still retain their proper stain. But if you've under-rinsed, you can also have gram-negative cells that appear to be gram-positive, and likely no effect. And this can lead to some issues if you're trying to identify an unknown bacteria. You need to be sure that you're ethanol rinsing for the right amount of time. Okay, so if you get a mixed result, what can you do? Well, this is why in our lab we're going to run controls whenever we do this. We need to have a gram-positive control and a gram-negative control, because this is going to ensure that your staining your staining technique is on point. Okay, because if your gram-positive cells are going to be purple and your gram-negative cells are pink, then you can be sure that your stain in the middle is probably of your sample is going to be correct because your controls lined up. But you can also have things that happen with your intermediate results. If you have gram-positive cells but you over-rinsed, gram-negative cells but you under-rinsed. If you have an old culture, because the integrity of these cultures is reliant on the media that is present to them, this can also affect the viability of the cells, and their gram stain can then appear intermediate. And by intermediate gram stain, I mean they can appear both purple and pink. Or you can have a contaminated culture, and again, if you have proper controls, you can assess if this is true or not. Or you can have too many cells on the slide. If they're too clumpy and they're all dispersed in the same place, you're going to not have the stain or the ethanol penetrate correctly, and this can also result in some intermediate staining. Okay, so just an example. So what conclusions can you make about this particular slide? Well, you have your gram-positive cells that stain correctly, but your gram-negative cells appear to have some purple present. So this indicates that you probably didn't decolorize enough and that some of these cells are still present. Or you have a gram-negative sample that's contaminated. So you can't really assess what's actually happening with your sample. Okay, so keep that in mind when you're doing your stainings for the gram stains. Now this is an example of what you would expect if something correct is happening. So you have a gram-positive control that's great. Your gram-negative cells are pink, as they should, and your sample has intermediate coloring. So, so from this, you can assess that this is probably a good